So last time we were talking about introns, how they need to be spliced out of the messenger RNA transcript. Otherwise, they'll include a lot of extra codons, which code for a lot of extra amino acids. And we said that there was a protein structure called the spliceosome, which involves these small nucleoprotein RNA molecules, snRNPs or SNRPs. And so they help to cut out and remove the introns. And we said that bacteria do not have spliceosomes, so bacteria are not capable of removing eukaryotic introns. Once the introns are cut out, then we have the final functional messenger RNA, which, when properly translated by a ribosome, will produce a functional protein. So we saw yet another example of how RNAs can be catalytic. So the ribozyme is an RNA molecule that acts like an enzyme. So RNAs can be catalytic. So all biological catalysts are not proteins anymore. They can be a protein or they can be an RNA. And we talked about alternative RNA splicing as a way to generate multiple messenger RNA transcripts, each coding for a different protein, but they're all coming from the same gene. So when the introns are spliced out differently, you get different messenger RNA transcripts which code for different proteins. So even though I said that humans have approximately 30,000 genes, we produce more than 30,000 proteins because some of those genes have introns that are alternatively spliced. And that produces different messenger RNAs and different proteins. And now the Human Genome Project, where they have completely sequenced the human genome, most genes with introns are alternatively spliced. So again, we make a lot more proteins than we have genes. So that's something that prokaryotes cannot do, but eukaryotes can. So one gene, but more than one protein or more than one polypeptide will be formed from that particular gene. Yeah, question, Chris?
Hey, Professor, are the introns, are they always spliced out whole? So it's just which ones are spliced out, or are they parts of them are spliced out to make the difference?
If they're alternatively spliced, then parts of them are spliced out. Okay. As we see here in this example, where we have two introns, maybe one gets spliced out, but the other doesn't get spliced out, or maybe half of an intron gets spliced out, but not the other half. So there are different types of mRNAs that can be produced, but depending upon how the introns are spliced out. What mechanism controls that? I have no idea. But introns can be spliced differently and produce different messenger RNAs. So this is one way that eukaryotes can evolve new proteins. They don't have to change the gene, right? So they don't have to accumulate mutations. They can just splice out the introns differently and produce a new protein. Now, introns are also the locations where the adjacent sister chromatids touch during meiosis and where crossing over occurs. So that way you're not, cutting a gene or an allele when crossing over occurs. It occurs where there is an intron. That way, if something happens and maybe the intron isn't cut perfectly, it doesn't interfere with the genes that are on that sister chromatid. So wherever you have the introns, that increases the probability of a crossover event. What did we call it where the sister chromatids touched each other for crossing over? What's the fancy word we use for those places where they touch? Remember chiasmata? So very often there's an intron there, and that's where this adjacent sister chromatids touch, and the pieces break off and switch places. So it happens at an intron. So the presence of introns allows for the genes to get mixed up more during the formation of eggs and sperm. And that again increases variation, increases diversity, which enhances adaptation and survival. All that leads to evolution. So this is yet another way that eukaryotes can increase the variation in their genomes. Because they have introns, crossing over will occur, and that allows the adjacent sister chromatids to switch pieces, which increases the diversity in the gametes. So it mixes up the genes more. Because the whole process of crossing over is a risky process, right? Where the adjacent sister chromatids pieces have to actually break and then switch places and reattach. Sometimes that doesn't happen. Sometimes one of the pieces that breaks off a sister chromatid floats away and is lost. So that will create a defective gamete, or at least a gamete not carrying enough genes. And we'll see some of those problems next week. So all this risky stuff, splicing out introns, if it's not done correctly, then that messenger RNA is not going to code for the right protein, but it can be done alternatively to produce different mRNAs and different proteins. So this all enhances variation. All right, so now we're going to talk about translation or protein synthesis. Let's see how the ribosomes do that. So there's another RNA molecule that is very important here. Those are the transfer RNAs, or the tRNAs. Ribosomal RNAs, the rRNAs, are a structural component of the ribosome. As it turns out, ribosomal RNA is probably the most important component of the ribosome and allows for the catalysis involved in joining amino acids together to form that polypeptide. So again, we're seeing another example of where an RNA molecule is a catalyst. So there's a specific transfer RNA molecule which carries only one particular amino acid. And that tRNA molecule has a complementary sequence on it that is complementary to the codon on the messenger RNA. So we call that complementary sequence on the tRNA the anticodon. So the anticodon is a complementary sequence on the transfer RNA which matches the complementary sequence in the messenger RNA. So that way, all the tRNAs, or the ribosome knows which tRNAs to allow in to attach their amino acids in the right sequence. So for example, if the codon on the messenger RNA were UUU, the tRNA would be AAA, right? That's complementary to UUU. So it's the same base pairing rules, except since we're dealing with just RNA, there's no T, it's a U. But otherwise, it is a complementary strand written the way that you've done before. Now, the anticodon is also the same sequence in the original DNA molecule, except that there's no T, because a tRNA is RNA, so no thymines, but it does have uracil instead. So if you were ever asked, what's the anticodon from a DNA sequence, it's the same as the DNA sequence, except that if there are any T's in the DNA sequence, you would just change them to U's. And so that's the anticodon. It's just like the original DNA sequence used to make the messenger RNA, except that there's no thymine. It's uracil. So the RNAs, ribosomal RNA, tRNA, messenger RNA, they're all synthesized in the nucleus of a eukaryote, right? Because the DNA is in the nucleus, and the DNA is used as a template for transcription to make RNAs. So all the RNAs are synthesized in the nucleus of a eukaryote. But there is no nucleus in a prokaryote, right? So this all happens in the cytoplasm of a prokaryote. So what does a tRNA actually do? Well, it picks up its amino acid from the cytosol, from the cytoplasm, and there's always an available pool of amino acids floating around inside the cell. And then the tRNA takes that amino acid to a ribosome. The anticodon will match with the codon in the messenger RNA, and the ribosome knows it needs to connect this new amino acid to the growing polypeptide chain. And that tRNA will float away and pick up another amino acid. So here we see a tRNA molecule. It's only about 80 nucleotides long. It does fold on itself, so it can base pair with itself. This makes the tRNAs a little more stable, right? Because we already talked about how RNAs are not very stable molecules. If it folds on itself and base pairs with itself, that enhances stability, so the tRNAs will survive, for a while inside the cell. Otherwise, the cell has to constantly create brand new tRNAs, very, very frequently. So it doesn't want to do that. So tRNAs hang around for a little bit before they need to be replaced. Notice that there is an anticodon, which is complementary to the messenger RNA codon. Here's a bit more realistic structure in terms of the folding of a tRNA. And for the sake of simplicity, this cartoon, sort of tube, a bent cylinder is what they use to show the tRNA, just because it's easier to draw this than to draw either one of these. So again, the anticodon is complementary to the codon on the messenger RNA. There is an attachment site for the amino acid. If each anticodon had to be a perfect match to each codon, we would expect to find 61 types of tRNAs, but the actual number is only about 45. And so the anticodons of some tRNAs can recognize more than one codon. And that's because we know that for some of the codons, there's multiple codons for the same amino acid. Remember how last time we looked and said things like leucine had 6 different codons, and 4 of them started with the same first two nucleotides, and the third one, could be any other nucleotide. Well, same kind of thing. The tRNAs, they can match the first 2 nucleotides, and the third one is flexible, and so they call that the wobble. And so that's just so they don't need to, a cell does not need to synthesize 61 different tRNAs, one for each codon. Only about 45, so that cuts down on the number of tRNAs required. So now this animation is one that on the very first slide, I said that you could Google it to see if you could find it, the CPUP protein synthesis. It does work on my laptop, so maybe it'll work on yours, but I'll just go ahead and show you this right now. I'm going to share my screen. So this is the CPUP protein synthesis animation. So it has some explanations to go along with it. So it's a nice thing to review the process, right? It tells you things like transcription is the first stage in making a protein. Right, during transcription, the information coded in the cell's DNA instructs the cell to make a messenger RNA molecule out of individual nucleotides. And transcription takes place in the nucleus. So here it's showing you the RNA polymerase enzyme, right? The DNA has been opened up. Only one of the strands will be used as a template to synthesize the messenger RNA. And remember, since it's RNA, there's no T in RNA, it's U. So no thymine, it's uracil. So the RNA polymerase is starting to synthesize, right, the messenger RNA. You notice that it started with what sequence? What was the first codon that was made there? You got to talk to me when I'm sharing my screen.
AUG.
Right, AUG, right? And that'll be used as the start signal for the ribosome later. So once transcription is complete, right, the new messenger RNA strand will leave the nucleus and move into the cytoplasm. Now, remember that there's a lot of modifications that have happened before it leaves the nucleus, right? A 5' guanine cap, a poly A tail, introns getting spliced out, and then it travels to the cytoplasm. So then it will hook up with a ribosome. And as we'll see, the ribosome is made of two subunits, a large subunit and a small subunit. They actually do not come together to form a ribosome until there's a messenger RNA there to be translated. Right? So the ribosomes exist as two pieces and only come together when there's a messenger RNA in order to be translated. And so each tRNA, and that's shown by these things that look kind of like jellyfish. And so we see the anticodons here. Well, they are complementary to the codons in the messenger RNA. And that's how the ribosome knows which amino acids to put in the correct order. Now, the way this works, if you were playing with this on your own, you would grab the tRNAs and put them there. So AUG is our codon. What is the anticodon for AUG? Remember, there's no T's in RNA. What's the complement to AUG?
UAC.
UAC. So if you look around, you'll find, oh, here's a UAC. Bring it here, and it moves to the next codon. GUA. What's the anticodon for GUA?
CAU.
CAU. So I'll grab that one here. Now what happens if I grab the wrong one and try and stick it in there? It won't do it. So CAU is our next codon. What's the anticodon for CAU?
GUA.
GUA, right? AAA, that's the easy one. Right, because we just saw that in the lecture. What about ACU? Anticodon for ACU?
UGA.
GUU? What is it? CAA, right? What about UGC? UGC.
ACG.
CAC? GUG, right? And GGU, BCCA, ACA, UGU, UUG, AAC, AAC, UUG, and UGU. And UAA is a stop codon. At that point, the ribosome will break apart, right? The large and small subunits will separate, and the messenger RNA will float away too. So then we have our polypeptide chain. It's not technically a protein until it's all folded on itself. And there may also be some post-translational modifications that we're going to talk about next. All right, so a nice little animation to illustrate the process. But it doesn't take me to where we were. I got to get back to where we were. Now, in order for a transfer RNA molecule to actually pick up an amino acid and take it to the ribosome, energy is required to attach that amino acid to the transfer RNA. And so there is an enzyme which does that called amino acyl tRNA synthetase. And you notice that ATP is required in order to join the amino acid with a transfer RNA molecule. Since we have 20 different amino acids, we have 20 different tRNA synthetase enzymes, one for each amino acid. So energy is required to attach the amino acid to a transfer RNA molecule. So here we see the ribosome is composed of a large subunit and a small subunit. They come together when there's a messenger RNA there. There's also ribosomal RNA that's part of this that actually does the catalyzing, where the nucleotides the amino acids get added together to form that polypeptide chain. And now the RNAs are synthesized in the nucleus, and the RNA and the proteins for its subunits in the nucleolus or the nucleolus. So it's just a region within the nucleus where the RNA molecules are synthesized along with the ribosomes. And then all the subunits exit the nucleus and enter the cytoplasm. And when a large and small subunit of the ribosome come together, you have a functional ribosome, but that doesn't happen unless there's a messenger RNA there. Eukaryotes and prokaryotes perform translation in the same way, except that the ribosomes are a little bit different. And that difference is enough that we can exploit that difference. There are many antibiotics which we use to treat bacterial infections that interfere with protein synthesis in bacteria, but not our cells, because the ribosomes of bacteria are different. And like I said, that's a difference we can exploit. And we can find drugs to target the prokaryotic ribosome and kill bacteria because they can't synthesize their proteins if we're taking that drug. So the overall process is the same, but the ribosomes are just different enough that we can exploit that difference with drugs and target translation in bacteria and kill them without harming our cells. And tetracycline is an example of a broad spectrum antibiotic. And when you go on to take micro, if you take micro, you'll learn all about these things in micro. So on the ribosome, there are 3 sites: the P site, the A site, and the E site. So it looks like the EPA. So the P site holds the tRNA that's carrying the growing polypeptide chain. The A site carries the tRNA with the next amino acid that's going to be joined. And then a discharge transfer RNAs leave the ribosome at the E site. So it's kind of like an assembly line. As the ribosome is scanning and reading the messenger RNA, tRNAs come along, and the correct one with the anticodon will land into the A site, and its amino acid will be joined with the other amino acids in the polypeptide chain. So we see the polypeptide chain here. Here's our next tRNA carrying an amino acid. What actually happens is the chain is added to the new amino acid rather than the new amino acid being added to the chain. It's the chain that's actually added to the new amino acid. So once again, this is just emphasizing the point that the ribosomal RNA molecule that's part of the ribosome is what performs the catalysis, is what forms the peptide bond right between the amino acids to form that polypeptide chain. So translation can be divided into 3 stages: initiation, elongation, and termination. All 3 phases require protein factors that aid in translation. So there are a lot of protein factors, and we've talked about these before. Right, with DNA replication, we know that there are many enzymes and DNA binding proteins involved in the process of replication. We know that with transcription, right? The RNA polymerase, it binds to the promoter sequence, but there are transcription factors that enhance the binding of the RNA polymerase, right, with the promoter. There are also transcription factors that inhibit transcription. When it comes to translation, there are more factors that are required to enhance translation. So all these different protein factors are involved in the regulation of gene expression. So the formation of ultimately a protein from a gene involves many levels of control. And so just because you possess a particular allele of a particular gene does not mean you will express that allele. There are many factors involved in gene expression. And so here we're just talking in general about all these protein factors, but they affect the formation of proteins, which means they're affecting the expression of a gene. So regulation of gene expression in eukaryotes is very complicated, right? There's not any one simple mechanism for eukaryotic genes. In prokaryotes, it's much simpler. In the last lecture, we will talk about ways to regulate gene expression. In bacteria mostly, and we'll mention a little bit about how some genes get turned on and off in eukaryotes. But the whole process involves many parts and pieces. So that makes it complex. It also creates more possible places where a mistake can happen. Whenever you have a lot of parts and pieces that all need to work perfectly, if one of them doesn't, then that causes problems. It messes up the whole system. So on the one hand, it's remarkable that DNA replication, transcription, and translation happen as well as they do. But every now and then, there's a problem somewhere. So just keep in mind all these different things that are required to make something happen. If one of them isn't working right, if one of them is missing, it fouls up the whole process. So it's amazing how well everything normally works, but sometimes it doesn't. Now, both initiation and chain elongation require energy. Here, GTP, or guanosine triphosphate, is going to be used instead of ATP. So it's basically the same idea. When you hydrolyze the GTP, that energy, it's an exergonic reaction, and you're coupling it with an endergonic reaction to make the whole thing exergonic and therefore spontaneous. So same idea here, except instead of ATP, GTP is used. But the same idea. So initiation and elongation require energy. Adding the amino acid to a tRNA requires energy. Synthesizing all those tRNA synthetase enzymes requires energy. So protein synthesis is an energy-intensive process. It takes a lot of energy to make a protein. You got to synthesize enzymes to make it happen. You have to use ATP or GTP in order for the ribosome to be able to do its job. So making a protein is energy intensive. It requires a lot of energy to make a protein. And proteins may be hundreds of amino acids, long. There may be hundreds or even more amino acids that comprise a particular protein. So a lot of energy goes into making proteins. So GTP is the energy source for protein synthesis, also for gluconeogenesis. Does anybody know what gluconeogenesis is? the name kind of tells you, if you can figure that out, but gluconeogenesis. Any idea?
Production of glucose sugars.
Right, from non-carbohydrate sources. So there are some amino acids which can be converted into glucose and then used for aerobic cell respiration. So gluconeogenesis is something where GTP is mostly used to make glucose from non-carbohydrate sources. So initiation will bring together the messenger RNA, a tRNA, with the first amino acid and the two ribosomal subunits. So the ribosome does not assemble unless there's a messenger RNA present. Then the large and small subunits come together. And guess what? That requires energy. Right? So putting together the ribosome requires energy. And then that first tRNA, which of course is carrying methionine, because AUG is the start codon, and it codes for methionine. So all of our polypeptides start with methionine, and it lands in the P site. And for elongation, other tRNAs will come along carrying the appropriate amino acid, and they'll be joined together. And again, this requires energy, right, for elongation. So here we have the assembled ribosome. Notice that this tRNA has a series of amino acids joined together already. Our next tRNA comes along with its amino acid. Then the polypeptide chain is actually transferred to that new tRNA, right? So the amino acid that's being carried by this new tRNA isn't, given up to the polypeptide chain. The polypeptide chain is joined to it, right? So that growing chain is added to every new amino acid, as opposed to every new amino acid being added to the chain. So it's a fine point. It's not a big deal. And so then the tRNA that was in the P site moves to the E site and leaves. And then that new tRNA moves over to the P site. And then another tRNA will come, land in the A site, and the polypeptide chain will be attached to that new amino acid. And then the previous tRNA will slide over to the E site and leave. And then the newer tRNA moves over to the P site and makes room for the next tRNA to come along with its amino acid. And this process, it just repeats over and over and over again until a stop codon is reached. When a stop codon is reached, then the ribosome falls apart and releases that messenger RNA. But this whole process requires a lot of energy. So remember, there are 3 stop codons. And once the ribosome hits one of those stop codons, everything falls apart. And there's yet another protein, a release factor, right, which helps that happen. So again, there's a lot of these protein factors involved in all these different processes. So it's actually more complicated than what we're discussing. We just want to focus on the main ideas with a few details. So we have another animation, and this is a YouTube one. So again, I'll share my screen. And we'll watch this short video animation. I'll turn down my volume.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site. Translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
So, a nice little summary. Any questions? So a single messenger RNA can actually be translated many times. So remember when we were talking about transcription, how one RNA polymerase molecule can hop on and start transcribing to make a messenger RNA, and then another RNA polymerase enzyme hops on right behind it and starts synthesizing another messenger RNA, and then another RNA polymerase can get on behind that, so that a gene can be transcribed multiple times by multiple RNA polymerases. But the same kind of thing happens here with translation, where more than one ribosome can attach to that messenger RNA and be translating it. So we call these polyribosomes, and you can see in the electron micrograph here all the different ribosomes that have attached to this messenger RNA molecule. And so one ribosome after another will attach to the start, and then they'll read the messenger RNA, and another ribosome can attach to the start and continue to read, and so on and so forth. So if cells need to make a lot of a particular protein, They can have multiple transcription producing multiple messenger RNAs, and then those RNAs can have multiple ribosomes, all translating the same messenger RNA, so that a lot of protein can be produced pretty quickly. A ribosome requires less than a minute to translate an average size messenger RNA into a polypeptide, so relatively quickly. And if you got a bunch of ribosomes all translating simultaneously, you can make a lot of proteins very quickly. Now, as the polypeptide is being synthesized, once it gets to near the end, it will start to fold on itself. And remember, this is the tertiary level of protein structure, when everything kind of folds on itself and makes all those different kinds of bonds, like disulfide bridges, hydrogen bonds, van der Waals forces. And so that could be it for that polypeptide. It now folds into a functional protein. Some proteins are more complicated, and they need chaperone proteins to help it fold correctly. And there are certain proteins, if they're partially denatured, they can sometimes renature, refold with the help of chaperone proteins. But if a protein has been completely denatured, it's most likely not going to be able to ever refold. Now, some proteins will require post-translational modifications. A glycoprotein needs to have a sugar added onto it. A lipoprotein needs to have a fat molecule attached to it. So there are different kinds of modifications that can occur. And we know from the quaternary level of protein structure that more than one polypeptide subunit needs to associate together to form that functional protein. We find two types of ribosomes in the cell. We have free ribosomes, which are in the cytoplasm, and then we have the ribosomes that are attached to the endoplasmic reticulum. And remember, we called that the rough ER because it has ribosomes attached to it. So some messenger RNAs, they just go to the cytoplasm to hook up with a ribosome and be translated there. Others will go to the endoplasmic reticulum. And so that polypeptide, as it's synthesized, moves into the ER. While inside the ER, it may receive those modifications like having a sugar or a fat added onto it. There is also a signal peptide associated with the polypeptide, which helps to tell the cell where to deliver that protein. Sometimes those proteins are destined to be secreted to the exterior of the cell, so we call those secretory proteins. Sometimes these are integral membrane proteins, so we know that there are proteins that span the cell membrane, and so they will be embedded in some of the ER's membrane, which will then bud off and form a vesicle, which ultimately fuses with the cell membrane. And now you've added those transmembrane proteins to the cell membrane. But there are signals which tell the cell where to deliver all these different proteins. So here we see the rough ER, with the ribosome attaches to the rough ER, and there is a pore, right? There's an opening. As the polypeptide is synthesized, it moves into the ER. There, it can be post-translationally modified. It will be packaged in a vesicle, which will bud off and head towards the Golgi, right? Because the Golgi was kind of like the post office that delivers all the proteins throughout the cell. And so the peptide signal is like a postal code, or we would call it a zip code here, which helps to tell the cell where to deliver these different proteins. So again, we see something that is very complicated, where all these different proteins serve different functions, and they have to be delivered to different places inside the cell. And the way the cell knows how to do that is with a signal peptide. Again, how the cell knows which signal peptide is which, I don't know, but the cell does know where to deliver all these different kinds of proteins. So the point here, RNA plays multiple roles inside the cell. It's not just messenger RNA, it's tRNA, it's rRNA. They have small RNAs, which are involved in splicing out the introns. RNAs are catalytic. And as we'll learn next week, there are some RNAs that modify gene expression. So RNAs do a lot of different things inside the cell. But the big one is the messenger RNA that we always talk about. But you can't translate the messenger RNA without transfer RNAs and ribosomal RNAs. But there are many other RNAs that are in the cell that perform a variety of functions. So DNA may be the genetic material of all living cells, but RNA is more versatile because it does a lot of different things, including the regulation of gene expression. We'll talk about that next week. So lots of different kinds of RNAs are involved. Now, once again, the prokaryotes, they do transcription and translation in basically the same way, but there are some differences, especially when it comes to the regulation of transcription and translation. There are different RNA polymerase enzymes for eukaryotes and prokaryotes, so they're not the same. When it comes to the termination of transcription, remember that for prokaryotes, when that terminator sequence is reached, the RNA polymerase just falls right off. But for eukaryotes, the RNA polymerase kind of continues past at that terminator sequence for a little while. And we mentioned how the ribosomes of prokaryotes are different than eukaryotes. And that's a difference we can exploit to kill bacterial cells without harming our cells. We've already discussed how bacteria can make a protein faster because transcription and translation can occur simultaneously in a prokaryotic cell. In a eukaryotic cell, transcription occurs in the nucleus and translation in the cytoplasm. So the messenger RNA not only has to be modified, right, the intron spliced out, a 5' cap, a poly A tail, then it has to be transported from the nucleus to the cytoplasm, where it can then be translated. But for prokaryotes, no nucleus. Everything is happening in the cytoplasm, so transcription and translation happen simultaneously. No post-transcriptional modifications are required to a prokaryotic messenger RNA. Right, so no 5' caps, no poly A tails, no introns. And so a protein synthesis is faster in a prokaryotic cell. And because prokaryotic cells are smaller, most of the proteins just diffuse through the cell to wherever they're needed. So no postal codes, no zip codes, right? So no delivery because there's no ER, no Golgi in a prokaryotic cell. So prokaryotes are simpler, but they can also do things faster and more efficiently than eukaryotic cells can. So this is just saying what I just said. All right, so now let's talk about some of the problems that can happen, and namely the problems when there are changes in the DNA sequence. And we call these changes mutations. Mutations happen with all things, including viruses, which are not technically living things, but we call them organisms. And so mutations occur when there's an alteration in the DNA sequence. And when it comes to mutations, there are three possible consequences. They can be good, they can be bad, or no difference, no change. They can be neutral. How can a mutation be good? I mean, whenever you heard that word mutation, throughout your entire life, when you watch sci-fi, mutations are always bad things. But sometimes they're good. How can they be good?
If the mutation results in a positive gene, like, I think there were those moths, and one of them mutated to blend in with their background better, and they survived more.
Well, there was natural variation in the population because of mutations, and then a selective pressure, like the birds being able to see the lighter moths more easily. They got eaten, and the darker moths were left behind, so that became the predominant characteristic. But that variation was there because of mutations. Some mutations create variation. And the outcome of a mutation or of that particular variation may not be apparent. It could be a mutation that doesn't do anything until the environment changes. And suddenly, those members of a species that possess that particular mutation, that particular characteristic, now they're better able to survive. So just because you have a mutation doesn't mean that it's going to be immediately apparent. And we all have mutations in our genome, so none of us is genetically perfect. Now, there can be large-scale mutations where long segments of DNA are translocated. They're moved from one chromosome to another, or one place to another place on the same chromosome. There can be duplications of sequences, inversions where a sequence gets cut out, and when it reattaches, it's backwards, and so it's not going to be transcribed or translated correctly. So there are many different things that can happen, but we're going to focus mostly on point mutations, normally single base changes, which change the codon. So a point mutation can produce different end results. So here we're going to look at the gene for hemoglobin. And even a single base change, it can change the hemoglobin from normal hemoglobin, and the normal we call the wild type, and then to a mutant hemoglobin, which leads to sickle cell disease. And we'll talk more about sickle cell when we do genetics. But a single base change produces the sickle cell trait. The same kind of thing with the most common type of dwarfism seen in humans is the result of a single base change in a single codon. And that's it. So one single base change, one codon change can have dramatic effects, but it depends on the protein and what kind of change it is that occurs. So a base substitution or base pair substitution, you can change a codon and still code for the same amino acid, right? Because the code is redundant. So it's entirely possible that a mutation occurs, which changes the codon, but you still get the same amino acid. So we call that a silent mutation or a neutral mutation, because nothing has changed in the final protein. But sometimes that base change will change the codon, and it now codes for a different amino acid. We call that a missense mutation. And that could potentially be very bad. And then a mutation that produces a stop codon is almost always bad. But we're just changing a base which changes a codon. If there's no change in the final amino acid, it's a silent or a neutral mutation. If we change the codon to a different codon that codes for a different amino acid, we call that a missense mutation. Whether that's good, bad, or indifferent depends on the nature of the amino acid that's changed. If you had an amino acid, for example, in the active site of an enzyme, and that particular amino acid was a non-polar amino acid, and the mutation changes that amino acid to one that's actually charged, either positively or negatively charged, that's going to repel the substrate when it tries to bind. And so now that enzyme is no good. It's not able to bind to the substrate and perform its function. Or maybe again, you have a change to an amino acid with different properties that prevents a protein from folding correctly because it can't make the proper, chemical bonds. Well, again, that protein's probably not going to be functional. If that's a critical protein, that organism's dead. A nonsense mutation, almost always bad because a stop codon gets produced, and that results in a shorter protein. It's missing, a bunch of amino acids. And so most likely that protein is not going to be functional. If that's a critical protein, that organism dies. It's possible, not likely, that you create a brand new protein which has such a wonderful new function that it helps that organism to survive. So a mutation could create a new protein with a wonderful function, or it could enhance the function of an existing protein and make it function better. So there are some mutations that are beneficial, and they help organisms to adapt and survive. But in general, most mutations are not good. So again, we see the wild type strain. If we have a silent mutation, as in the case here where our glycine codon GGC, it becomes GGU, but GGU still codes for glycine. So it's a silent or a neutral mutation. So there's no change in the final protein. A missense mutation occurs where you have a base change, which changes the codon, originally GGC, into AGC. That codes for serine. Serine is different from glycine. Serine has a hydroxyl group on it, an OH group. That could very well change how that protein folds or how it functions. And so if that's a critical protein, that organism will die. It could also enhance the function of that protein, right? In that case, it could be beneficial. A nonsense mutation is bad. A codon is turned into a stop codon, and so our protein synthesis will terminate prematurely, and you get a short protein that's not going to be functional. So it's extremely unlikely that a nonsense mutation will be beneficial. It's possible, it's just very unlikely. Insertions and deletions are also pretty bad because we're talking about inserting more than one, or in some cases just one nucleotide, but it could be more than one, or deleting one nucleotide, or deleting more than one. The problem when you have an insertion or deletion is that all the codons after the insertion or deletion are all changed. All the groups of 3 nucleotides, all those groups change. And that means all the codons change. So all the amino acids change after the insertion or deletion. So we call that a frame shift mutation because the translational reading frame is completely changed. So we just get a bunch of random amino acids. Sure, is it theoretically possible that a brand new protein gets created, which is wonderful? Sure. It's extremely unlikely, but it's possible. But it's extremely unlikely. So a frame shift mutation occurs where nucleotides are added or deleted, and that changes all the codons after the addition or deletion. So all different codons, all different amino acids. And if that's a critical protein, that organism is going to die. So any questions on these mutations? So here we see, again, the wild type, and then a frame shift mutation where the U nucleotide is missing, and so that changes all the codons afterwards. So you get all different amino acids. Randomly, sometimes when all the codons are changed, you get a stop codon that's formed, right? So that's going to mess up your protein as well. Now, if the addition involves 3 nucleotides, or 6, or 9, or 12, all the codons afterward are still going to be the same, right? They're not going to change. So if your additions or deletions involve groups of 3 nucleotides, it doesn't change the reading frame. All the other codons are still read the same way, but the problem is if you have an addition, you have extra codons, which means extra amino acids. If you have a deletion of one or more codons, you're missing amino acids from your protein. So even if you don't screw up the translational reading frame, you're most likely not going to produce a functional protein because it's going to have either extra amino acids or it's going to be missing some amino acids. And that can affect how the protein folds and functions. So these are the major types of mutations that can and do occur. Mutations occur spontaneously. Remember, we said that DNA polymerase is not perfect. We do not want it to be perfect. It has to make a mistake every now and then. Right? Those mistakes lead potentially to beneficial mutations. Right? They add to variation in a species, and that allows the species to survive and adapt. So spontaneous mutations can and do occur. And we want them to occur. Even though most of the time they're going to be bad, you got to take the bad with the good, right? There is no evolution without mutations. So even though most mutations tend to be harmful, every once in a great while there's a mutation that's beneficial. And that helps the species to adapt and survive. A mutagen is a chemical or physical agent which increases the rate of mutation. So this is not a good thing. We don't want a high mutation rate because that creates way too many problems too quickly. And so organisms don't adapt, they die. So we don't want too many mutations to happen. There's actually a sort of limit where a narrow range of ideal mutation rates. If the mutation rate is too low, no variation, no adaptation, extinction. If the mutation rate is too high, it's just extinction because there's too many problems that are happening. So there is, it's kind of like a Goldilocks range of mutation rate, that's ideal to allow species to adapt and survive. But mutagens increase that rate too much. A physical factor like X-rays or gamma rays, very high energy forms of radiation that do a lot of damage to DNA. UV light. We talked about how UV light causes thymine dimers. We have DNA repair enzymes which normally repair most of this damage, but if there's too much damage that's been caused, we can't repair that, so mutations occur. There are many chemicals that cause mutations as well. And so, unfortunately, we often get exposed to some of these, or some people do things deliberately, right, to expose themselves, like smoking cigarettes. So both the chemical and physical agents increase the rate of mutation. And that's going to be bad, because that's way too many mutations to deal with. And so they usually cause death. There are different ways to test for mutagenicity. One of those ways involves bacteria, right? You grow the bacteria in the presence of your chemical agent and see what happens to the mutation rate. So there are ways to test, but that doesn't guarantee that something tested with a low mutation rate is actually safe, especially when you test this on other animals. Right? So that's one of the problems with animal testing. Animals don't always respond the same way that humans do. And so you can test something in an animal, no problems with that test animal. But then you start using it in people and you discover all these problems. So, but there are ways to test for mutagenicity and carcinogenicity of different compounds. But there's no guarantee that's going to translate exactly to human beings. So, just to sum up, we're going to start talking about Mendelian genetics next week. But we know that genes code for proteins and polypeptides and RNAs. And we're going to learn next week exactly how genes are inherited, how characteristics may or may not appear in the offspring. We'll talk about mechanisms of gene expression, and we'll learn just how complicated things are, way more complicated than Mendel ever could have imagined. But we know that a gene now codes for either a polypeptide or an RNA, right, molecule. And this image here just kind of summarizes the whole process of transcription, modifications, then translation in order to make the protein. A lot of parts, a lot of pieces. When everything comes together and works beautifully, it's amazing. But there are problems that can occur in many different places. So there are many different genetic diseases. We'll talk about some of these when we do genetics and see that as well as things usually work, sometimes they don't. And sometimes it's very bad. So a gene is a region of DNA whose final product is either a polypeptide or an RNA molecule. The regulation of transcription and translation is complex. Many transcription factors which stimulate or inhibit transcription, many translation factors required for translation to occur, and a lot of places where regulation occurs. So even if that allele is there in your genome, there's no guarantee that you're going to express it. It depends on a lot of different factors. And we'll talk about some of those. Well, we've already talked about some now, but we'll talk about some more in our last lecture for the course when we get to it. So questions or comments? I know there's a lot of stuff here.